Hello students, welcome to Learner's Planet. Let us continue with the next session of the chapter Fundamental Unit of Life. Now we have studied much more enough about this basic unit, that is the cell. We have studied about the basic characteristics, which are the similarities and the differences which exist in the different type of cells in the different organisms or within the organism. In the previous sessions, we have studied about the parts of the cell, the basic components. And in the last session, we have ended up with the prokaryotic nucleus and the function of the nucleus. So now, in this particular session, we are going to continue to study about the parts of the cell. We have done the plasma membrane, we have done the cytoplasm, we have done the nucleus. Now after nucleus, the next part we are going to study is the endoplasmic reticulum. So what is this endoplasmic reticulum? This endoplasmic reticulum is basically, it is a network of some tubular structures, and they are actually present all through the cytoplasm. They are present within the cytoplasm, that is for sure, but these tubes, which are scattered or which are present, they are not the independent tubules. Rather, each tubule or each section, each part of this endoplasmic reticulum is interconnected with each other. And you can imagine if so many tubes which are arranged in the cytoplasm and they are interconnected, so they can form a sort of skeleton and therefore forms an intracellular skeleton for the cell, and this it helps in maintaining the shape of the cell also. Now these tubes which are present here, they all are membrane bound. Now all tubes, whatever is present, they all are membrane bound. What does it mean? It means that it is a membrane bound organelle. The interior part of the endoplasmic reticulum is separated from the cytoplasm due to the presence of this barrier or a membrane in between. So although it is present in the cytoplasm, but still it is separated from it because of the presence of this boundary here. Now this membrane bound structure, it arises or it extends from the nuclear membrane and it continues up to the plasma membrane. See how strong is the network. Endoplasmic is it is entirely connecting each part of the cell. With what? With the nucleus and with the boundary of the cell. So these structures are they are actually membranous structure, and interiorly they are having the space. Therefore, you can see these membranes which are present in these structures, they are actually functioning as an envelope, and inside the envelope there is a presence of a cavity in which the various materials are filled. And that we will study in the further part of this session only. Before we switch to that, clear one more important thing, and that is that this endoplasmic reticulum, it is absent in a prokaryotic cell. Strictly, it is not present in the prokaryotic cell. So over there, the function will be performed by different parts of the membrane itself. Which membrane? Plasma membrane, or the components which are present in the cytoplasm over there. In the case of eukaryotic cell, there are certain cells which lack this endoplasmic reticulum. And you know what that cell is which is not having the endoplasmic reticulum? It is red blood cell, RBCs. RBCs of what? RBCs of mammals. You know what is mammal? Mammal is a kind of organism. It is a group of organisms which can feed its young one with the breast, as well as those who can give birth to the young ones. So these two are the special characteristics of the mammals. Now of course mammal is a multicellular organism. If it is a multicellular organism, then they must be composed of a eukaryotic cell. So all the eukaryotic cells, they contain the endoplasmic reticulum, but still the RBC is the one example which is devoid of endoplasmic reticulum. Structurally, the endoplasmic reticulum, it is comprised of so many tubular structures, and that we have already mentioned in the previous slide. Now these membrane-bound structures, either they are present in the form of tubule or they may exist in the form of vesicle. Now these two things should be very clear, that what do we mean by these two terms. Tubular structure is very clear, that is in the form of a tube. So it is something like this. So these are forming the tubules here. So they may be present in this way, like. So these are basically what? These are the tubules. But sometimes these structures, they may exist in the round sac-like structures. Now this outline which we have drawn here, it is actually resembling a membrane. Now you remember what is the structure of a membrane? The membrane, whether it is a cell membrane or it is a plasma membrane, it is made up of lipids and proteins. So these proteins which are present in the endoplasmic reticulum, they are different from those which are present in the plasma membrane. Why it is so? Because you can imagine the function of these proteins which are present in the membrane. They function as a transporter molecules or a carrier molecule or the receptor of various signals. So the signals which are supposed to be received by this endoplasmic reticulum is supposed to be different from that which has to be recognized by the plasma membrane. So therefore, although it is a protein, but it is somehow different from those which are present on the plasma membrane. Besides, they are made up of lipids, and these lipids, they are forming a bilayer structure. Remember what is bilayer structure? The lipid molecules, they are having a head structure and they are having a tail-like structure. This is a single molecule of a lipid. Now these are actually arranged in a particular fashion. This is one layer of a lipid membrane and the another layer is formed in this way. So further, this is forming a double bilayer structure. Now this bilayer structure, either it will form a tube of this sort or it will form this circular round structure. So what will be the structure now? It will be continued in this particular way. This is one layer and this will forming a second layer. Now you can see, if they are arranged in this way, either it is a tube or it is a circular vesicle. In both cases, the interior space is actually hollow. You can see this empty space in between, in the vesicle or in the case of this particular tubule. Now this particular space present here is known as a lumen. Let us understand with the help of this structure. Now we said that endoplasmic reticulum, it is membrane bound tubular structure which extends from the nuclear membrane till plasma membrane. So this interior part you can see here, this inner violet color structure is actually a nuclear membrane, and in between you can see these pink colored structures, these are the nuclear pore. Now in between they have formed a extension. The nuclear membrane will extend and will form a tubular network here. This tubular network is actually nothing but the endoplasmic reticulum. Now you can see these round structures which are bound here, and in some regions you can see even they are not present. So this we are going to study definitely now. But before that, just clear one thing. In these tubules, you can see they have formed a tube here. and in between there is a space. This space is known as a cisternal space, or it is known as lumen. So this particular lumen, or we can say a cisternal space, they are actually not empty. The lumen is filled with some fluid inside. A fluid is completely covering this particular cavity. Now what this fluid contains? The fluid contains water. The maximum proportion is water. So it is forming an aqueous solution inside. This aqueous solution, if it is a solution, then definitely it is not a plain water. It must contain certain other part also. That means the solute. So what those solutes are? They are the biomolecules or the biochemicals. Which biochemicals? They are proteins. Which type of proteins? Which are maintaining the structure of the membrane and the internal part of the endoplasmic reticulum fluid. Now these are further having some different kind of amino acids. Now these amino acids, they may be required for the proteins itself. They may be needed for the to produce a new kind of proteins. Besides, they may be synthesized by the endoplasmic reticulum itself, or it may be transported from cytoplasm or from other part of the cell to the interior of the endoplasmic reticulum. Now some of these proteins, they can function as the enzymes. And which type of enzymes will be present? Which are actually controlling the metabolic reactions taking place inside the endoplasmic reticulum. Now generally, these are the site for the synthesis of certain different amino acids. So the enzymes which are needed for such type of reactions, they are present within the lumen of the endoplasmic reticulum. Not only this much, different kind of products are also synthesized by ER, and that would be controlled by the enzymes. Now besides that, they need energy, and that would be provided by the carbohydrates. So another component present inside the lumen is carbohydrates. Besides, they contain lipids, minerals, and so on. Now these lipids and the minerals, of course, they help in the structural maintenance, and the minerals functions as the growth regulators. Now whatever biochemical here which is present, they are essentially required in a particular proportion for the maintenance of the ER as well as for the functioning of this endoplasmic reticulum. Now these membrane bound structures, they contain certain particles over its surface, and on the attachment of these type of structures or the particles, they can make its surface smooth or the rough. If they are absent, the membrane appears to be smooth. If they are present, the membrane appears to be rough. And on this basis, they can be of two types. The ER, it is of two types here. What is that? Either it is a smooth endoplasmic reticulum or it is a rough endoplasmic reticulum. The one in which the surface of the membrane, the outer surface of the membrane is smooth, that is known as smooth endoplasmic reticulum. As we can see in this diagram, it is showing a picture of a smooth endoplasmic reticulum. You can see these are the tubular structures. They are the network of tubular structures which are interconnected with each other. In the center, they are having the hollow space, and this hollow space is filled with the fluid, and that is this particular cavity or hollow space is known as lumen. And of course, the structure externally, the surface is almost smooth, and this is because of the absence of these type of structures. Besides, you can see in this another picture, the surface is containing some attached round bodies here, and these round bodies are known as ribosomes. And if the ribosomes are attached over the membrane, then it is known as a rough endoplasmic reticulum. So smooth endoplasmic reticulum, it is smooth, it is because of the absence of the ribosomes. What are these ribosomes? The ribosomes are the structures which are actually involved in the synthesis of the proteins. Now these are the structures which are made up of two subunits, and these two subunits, they are detachable according to the phase in which it is present. Or we can say, we said ribosomes are involved in the protein synthesis. So the stage of this protein synthesis process will actually give an idea that whether the two subunits of ribosomes are attached or they are separated from each other. But of course, if they are detached or attached, they will be located over rough endoplasmic reticular membrane. In SER, it is absolutely absent. While if these are absent in the smooth endoplasmic reticulum, then of course surface is smooth, but can you think how it is going to affect over there? The effect would be something different. That means the ribosome is involved in the protein synthesis. So this function of protein synthesis could not be performed by the smooth endoplasmic reticulum. But the membrane, although it is lack of ribosomes, but it contains various other enzymes which are involved in the synthesis of different types of lipids. There are so many kind of lipids, and all these are actually synthesized by the smooth endoplasmic reticulum. The raw material is present within the lumen, or it is transported from the surrounding region, that is cytoplasm or other organelles. Energy provided by the glucose, and of course the enzymes which are needed further, they are located over this membrane itself. The lipids which are synthesized by it, they are actually involved in so many processes, so many functions of the cell. One of the most important function of this lipid is what? It is a major part of all cell membranes. We have studied since beginning that all the membranes, they are composed of lipids, and thus these lipids are actually, from where they are coming? They are coming from the smooth endoplasmic reticulum. The next type of endoplasmic reticulum is rough endoplasmic reticulum. Now why they are rough? We have already started this point. It is because of the presence of certain structures over its surface, and these are known as ribosomes. So the attachment of ribosomes will make the surface to appear rough. And of course, this roughness is not only the characteristic which is imparted by the ribosomes here. Rather, it is involved in the synthesis of proteins. And how important these proteins are, we have already studied. We said that the code for the protein is located over the chromosomes. In the nucleus, they contain the chromosomes. The chromosomes are having the genes. Each gene is coding for a particular protein. Now we are saying, if it is coded by the gene, then how it is synthesized by the endoplasmic reticulum. So don't get confused. What happens here is, in the nucleus, chromosomes are present and the genes are located over it. Gene is chemically a DNA. This DNA will form RNA in the nucleus itself. Now these RNA will be transported to the endoplasmic reticulum, and then here they will get attached with these round structures, that is ribosomes. So the RNA is transported from nucleus to the endoplasmic reticulum, and in the endoplasmic reticulum the ribosome will get attached and detached with the RNA. And as they get attached, they will read the code which is actually transported by the genes. So if the code is actually read properly, they will start attaching the type of amino acid which is coded by the RNA. Now this particular amino acid When it attaches, so many amino acids will attach together as per encoded information, and they will form a functional protein. So all type of functional proteins are now synthesized by rough endoplasmic reticulum. But this process is not possible if the nucleus is not giving the correct information in the form of its genes and in the form of RNA. So this is why the function is taking place in a interconnected manner. Now you can see that how this ribosome is involved in the process of protein synthesis. Now these proteins which are synthesized by the endoplasmic reticulum, they are now transported to the target site. See, this diagram is showing here, the lumen is there, ribosomes are there, and they are getting attached with these RNA here. And these RNA will form the proteins now. Proteins which are present here now in the endoplasmic reticulum, they will be transported to the next organelle, that is Golgi apparatus. From there the processing will start and it will be transported to the target site. Now these different proteins which are synthesized here, they have different kind of functions as we have studied in the previous session. For example, they can function as a structural protein or as a regulator, or they can function as an enzyme, or they can function as like hormones, or they can control the different kind of metabolic reactions. So the function of protein is unlimited. In the absence of any kind of protein, the entire function of the cell is going to be inhibited. Hence, the ER is playing such an important role. Whether it is smooth endoplasmic reticulum or it is rough, both are having equal importance. And of course, they are involved in the formation of the membrane. So the function or the role is not actually limited in any case. They are so important, and that's what we end up in this particular session. Next we will continue in the next session and will explain the entire functions of the endoplasmic reticulum. Till then, thank you so much. We'll catch you in the next session. Have a nice day.